chain, it's considered a large pre-B cell. At this point, it's time to test out if the heavy chain is functional, by seeing if it can bind to a surrogate light chain and get expressed on the cell surface. The surrogate light chain is made of two proteins, V pre-B and lambda 5, and it's basically something for the heavy chain to practice with until the real light chain is eventually made. The surrogate light chain and heavy chain are joined together and placed in a vesicle, and then that vesicle is sent up to the cell surface. At the cell surface, the surrogate light chain and heavy chain are placed near two additional chains, immunoglobulin alpha and immunoglobulin beta, which are called side chains. If the surrogate light chain and heavy chain successfully make a functional heavy chain on the surface of the large pre-B cell, these side chains send down a signal to the nucleus. The cell starts to rapidly proliferate, and each daughter cell is called a small pre-B cell. The small pre-B cells now begin rearranging the light chain, which means that the pre-B cells will all have the same heavy chain, but each will have its own distinct light chain. And there are actually two types of light chains, a kappa light chain and a lambda light chain. The kappa light chain starts rearranging first, and it's an iterative process. In other words, initially a pre-B cell might create a kappa light chain that doesn't bind well to the heavy chain. If that happens, the cell will either get a chance to iterate and try a different arrangement, or in some cases the cell might simply get killed off. Not allowing B cell receptors to be self-reactive is super important, because if these faulty or self-reactive B cells are released, then they might simply not work and they might attack self-tissue and cause disease. To identify the self-reactive B cells, there's an autoimmune regulator gene, called AIR for short. AIR is expressed in primary lymphoid organs and allows them to express antigens that are normally found all over the body, like the brain, ovaries, and pancreas. It allows these primary lymphoid organs to serve as a microcosm of the entire body, kind of like how the Epcot Center theme park in Florida is a microcosm of the world. So that allows the body to test out these newly formed B cell receptors in a safe environment. If a small pre-B cell binds strongly to a self-antigen, then it dies swiftly and silently by apoptosis. Whereas if a small pre-B cell binds with only intermediate strength to the self-antigen, then the small pre-B cell will try to rearrange its light chain in the hopes that it'll reduce the strength of its binding to a self-antigen. Only small pre-B cells that don't recognize self-antigens at all develop into immature B cells. Now, if the B cell does have a faulty B cell receptor or a self-reactive B cell receptor, then the pre-B cell tries again by splicing out more DNA segments from its light chain to generate a new V and J rearrangement. In fact, the cell will keep trying to do this over and over, doing more and more rearrangements, until it's gone through all of the V and J segments on the first chromosome. And then it starts to splice out DNA segments on the second chromosome. Once all of the V or J segment rearrangements on the kappa chain of both chromosomes have been tried, the cell will start trying to rearrange the lambda chain. And it'll keep iterating with the lambda chain as well, until it runs out of genetic material. Ultimately, if there's a rearrangement where the resulting light chain binds to the heavy chain, and the resulting B cell receptor isn't self-reactive, then and only then does the cell become an immature B cell. Now, to get into the blood, an immature B cell has to signal to the bone marrow that it has completed its development. Now, remember that the B cell already has a B cell receptor with a mu heavy chain constant region, which made it an IgM antibody. But the B cell now undergoes a process of alternative splicing, which is where some of the B cell receptors continue to use the IgM constant region, whereas some start using the IgD constant region. But all of the antibodies, both IgM and IgD, still have the same antigen binding specificity. The presence of the IgD constant region on some of the B cell receptors is a bit like a driver's license. It signals to the bone marrow that the B cell is a fully rearranged immature B cell that should be allowed out on the open road. Alright, as a quick recap. B cell development includes rearrangement of the heavy chain, rearrangement of the light chain, and making sure that the B cell receptor isn't faulty or self-reactive. Heavy chain rearrangement means combining the DJ segments and then the VDJ segments. And then the VDJ segment is joined to an IgM heavy chain, and then it's paired with a surrogate light chain before the cell proliferates. Each daughter cell rearranges its light chain by combining the VJ segments, first with the kappa chain genes and then with the lambda chain genes. If the B cell receptor isn't faulty or self-reactive, then it'll express the IgD constant region, which will allow it to be released into the periphery as an immature B cell.